it's another episode of Viral Weather on the Weather Network. Today, where did COVID come from? How do we kill it? How do we make vaccines? How do we make ourselves better? A series of guests. Rachel Schoutsen is here from the Weather Network. Roderick Slavcev from University of Waterloo is here. Qasim Bukhari from MIT joins us as well. I want to go to you immediately, Roderick. How do we even find these viruses?
Well, in a case where you would have a potential outbreak, or you're recognizing that there is a viral outbreak, we would look at, what are the potential serum or what could be collected from individuals that have this infection. And from there, it can be grown on whatever the tissue is, the human tissue, where it's suspected of being or suspected of expanding. And once we have the genetic material, that can be sequenced and then matched against databases to try to isolate and identify what exactly is that virus, and to also then be used for how potential vaccines could be generated against it.
So this one, which came out of Wuhan in China, it was something that had some similarities to other viruses, SARS-type viruses, but it had its own unique traits too?
Well, the best way to think of viruses is that there's a potential for exchanges of genetic material to be happening all the time. In this case, coronaviruses can be infecting in animals just as they are in humans. And there is a potential for a jump that can occur where you have a virus that has the potential to infect not only an animal, but also to be transmissible between humans as well. So you have that co-infection potential that's available. And as a result, then you have this ability for a new virus to start expanding within a human population.
And it was Canadian researchers who were able to first isolate that virus. So how important was that step in order to, start this research?
Oh, it's critical. I mean, that is a critical piece of it, because we have to know what we're dealing with first. And you can't really design a vaccine against something that you don't know what it is. So as granular that understanding as possible, that all goes towards the efficacy and, of course, safety pieces that would come into the generation of a new vaccine.
And it seems like we found it pretty fast. We can give Canada a pat on the back for this one.
Absolutely. And I mean, I think about this on a global level. I mean, this really is a global collaboration that's at work here. And to see, I mean, not to take away from the grimness of what's happening with COVID-19, but from a scientific perspective, what you're seeing happening here is this is a rich moment for science, where you see scientists really stepping up to do their best and to collaborate as necessary to get this done.
I have a question. Why so much attention on this? The last time this kind of attention was drawn towards, medicine was SARS, which was almost 20 years ago. But why the intense scrutiny and concern with this one?
Well, even SARS was relatively, contained into a small area. Here we really have global impact of a virus like this. So not to say that the same thing couldn't have happened in SARS in 2003, but here this is a global impact, and this is something that you haven't seen an urgency of a pandemic like this since Spanish flu. And this is really something that again forced the hand of the entire globe to rise to the occasion and to generate a new vaccine and to protect against this.
And when it comes to vaccines, Roderick, I know you're doing incredible work with the potential of a nasal spray.
Yes, so the nasal spray, I mean, has a few potential advantages, and not to say it's a perfect system in and of itself, but it is a way to try to mimic the infection. And what we're trying to do is to get as close to what a normal infection would look like through SARS-CoV-2, and to follow that same pathway, but doing it with very, very safe components in how we sort of mimic that procedure, so that we can generate the most pertinent and maximal immune response that would be protective for individuals.
So what's the difference between bacteria, germs, and a virus?
Well, I mean, if you call them germs, I mean, germs could be just about anything, I guess, anything. But we tend to give that term to something that's, has it's pathogenic. So, it could generate sickness in humans. Bacteria, not all of them do that. Some bacteria are good, or they're commensal and do really nothing. But a virus, again, it's microscopic, and it has the potential as a parasite to be able to grow in our cells and to obviously cause sickness and disease, the same way some bacteria, of course, could as well. The difference between them is that we could look at a bacteria as actually an organism. A virus is not really an organism. It would be kind of a genetic entity that can kind of hijack the cells of whatever its host is and propagate its own genetic material, and in often cases cause disease.
So it actually gets into your cell and alters it.
Yes, it gets into your cell. It often it has to be expressed in one way or another. That doesn't necessarily mean in your nucleus. It depends on what the genetic material is, and it really just takes your cell, turns it into a factory to really mass produce its own genetic material and its own products, so that it becomes a factory for more of the viruses to be produced.
And then it just overwhelms the human body. We're having a terrific talk about COVID-19 and where exactly viruses come from. We're also going to discuss how we're making out on creating a vaccine, how long it will be before that's available, how you can actually kill viruses, and is the weather helping us or not? So we've determined now exactly how we find a virus. Let's bring in Qasim Bukhari from MIT to talk about your work that you've been doing on the spread of COVID-19. Qasim?
Yeah, hi. Thanks, Chris. So what we did was that it was more of a data analytics study. We looked at all the countries in the world, so all the COVID-19 cases that we could find. The updated version includes around 3.2 million of COVID cases that have already occurred across the world. And what we did was that we started identifying patterns related to the weather in this data. So we looked at the local weather from all the countries where these cases occurred, and we started identifying that if we can find something.
So you were looking to see if there was like a correlation that like a certain type of weather would accelerate the spread and help propagate it, right?
Yeah, the original intention of the study was just to identify whether we can predict the spread anywhere. So that was not to look at specifically the weather-related patterns, but we thought that perhaps inside this machine learning model, it would be good to have, for example, how the weather affects this. So we started modeling the weather part of it. And once we started modeling it, we realized that perhaps the whole project is just too big to do it within a few weeks. So we stick to it for the moment. Hopefully in future, we can actually bring the whole model in where we can just say, okay, if the COVID-19 is spreading, how the numbers would go up in a certain country after 1 week, for example.
India is an interesting study because there's 1 billion people in India, and the weather there is warm and humid. And how did that turn out for India?
That's a very good point, actually. So initially, the first version, so we have been revising the whole paper multiple times. The first version that we put in was somewhere in mid-March. That time, India had not started testing. Obviously, we knew from colleagues there that there were not too many reports in the hospitals, but that's not any strong evidence when it comes to it. So we thought that, okay, maybe India is not really doing the testing, and that's why you don't see the numbers go up. Okay, that's a good, fair argument. But we saw that in the last few weeks, there were a lot of testing done there. Now it's over 1 million tests already done there, right? And the number of COVID cases are much, much, much lower. Obviously, India has a huge population, so 1 million or more than 1 million tests done with respect to the population is not a huge number. But then the number of COVID cases is so little, is not even 100,000, right? And each country that you pick in Europe would have 100,000 cases. So that's much, much, much lower. And again, compared to the population, it's actually a very small number.
Rachel, you were going to jump in. Yeah.
Yeah, and it's interesting because I've been following patterns with weather and a virus like influenza, for example, and there's been a lot of really interesting research. And after reading your study, I'm noticing that a lot of the things that you guys have found with COVID-19 and weather are similar to influenza. So, for example, that the virus does not travel as far in humidity. The virus tends to fall apart in heat. Is there anything that you found with COVID-19 that was a little different, that was a little bit more surprising than just what we found in correlation with influenza, for example?
So influenza is a good example, but there is even a closer example to the than influenza to the COVID, and that is basically SARS-CoV-1. So you see that this is SARS-CoV-2. So you have already seen in the scientific literature that SARS-CoV-1 has a temperature-related dependency, right? So you saw it already in the lab experiments. So it is very similarly behaving. So SARS-CoV-2 is behaving very similar in terms of its temperature and humidity dependency to its other relative, which is SARS-CoV-1. So you would have expected it, obviously. The other thing is that influenza has already gone through a pandemic and now it has become more normal. So people have developed immunity against it. So what it means is that it can obviously, because in certain weather conditions, it favors transmission. So you can get it, but you still have some kind of immunity against it. The problem here was that there is no immunity in humans against this virus because this is the first time we are encountering it. However, so that's why even if there is a little bit of a contact, you would get it.
Listen, if you're following along the podcast on iTunes and Spotify, the conversation continues in only seconds here. We're talking about COVID-19 and its spread vis-a-vis how the weather works with it. So we were talking about everyone's excited that maybe the weather in the summer when it turns warm and humid here in North America will kill this off. Qasim, will it?
Unfortunately, no. So we already have evidence from many warm and humid places. For example, one good example could be Malaysia, where it's consistently above 35 degrees centigrade, 40 degrees centigrade, very humid. However, there is still a lot of transmissions, especially there was one event that was a huge gathering, and that gathering actually infected around 1,000 people from one gathering only. So it means that the virus is still getting transmitted there even in such conditions. So it's not going to go away unless we are taking social distancing measures and other precautions that we should be taking.
It's interesting because, Qasim, I've read the MIT study that you've done and it connecting summer and COVID. And I found that there's a lot of connections that you guys have made to the weather in this virus that have been previously known. That, for example, in more humid conditions, the virus will not spread as far. Heat can help kill the virus or deactivate it, essentially dry it up. Was there anything that really stood out with COVID-19 that was different in the way it reacted to weather?
So, so far what we found was actually more similar to the other viruses, how they are behaving. I mean, if you just talk about the COVID-2, then it was more similar to the SARS-CoV-1 than any other viruses. And this is expected because there were some lab findings even for SARS-CoV-1 that showed already that there is a temperature and humidity dependency. So we do find this dependency, but this dependency alone does not mean that the virus is going to go away. So because we saw it already, that you may, again, there are multiple factors that are into play here. So the virus can transmit through multiple means. This one is also airborne, so it can also transfer through air. Perhaps in very humid places, the airborne transmission would be lower, but the other transmission modes would still be working. So you have to be careful.
One of the other things that's kind of neat about a virus is and Roderick, this is what I've known about viruses. The initial virus that infected people in Wuhan in China has mutated since that point, right?
Yes, that's correct. I mean, and that's what you would expect for a viral entity of this sort, is that, I mean, with every new infection, as it continues to propagate, there's a potential for mutations to start developing. So, of course, even in, for instance, the United States, that same virus that would be in that epicenter may not be the same that you'd see in Wuhan, and isn't. And very often, and as it relates to vaccines, those mutations could then lead to evasion of an immune response, which is, again, relating to vaccine, why it's so important that we can generate a vaccine that is able to have that cross-reactivity to protect against such genetic drift.
So is what the virus trying to do is it's adapting itself to its particular environment and evolving with it?
I wouldn't use the word adapting. I think the best way to say is that you're generating mutations at a rate, And the adaptation is really, if that helps the virus to propagate better within that condition, then those are the ones that go on to propagate. So that's a sort of selective pressure against it at that point.
Now, when it comes to the virus changing, does that mean the vaccines need to change as well? Is this going to be something where researchers are just constantly chasing their tail, trying to create that perfect vaccine?
Well, yes. I mean, to some sense, yes. And it depends on what type of vaccine you're using. We have different branches of the immune response. Some can be more cross-reactive than others. In a case like this, where you have a high mutation rate for a potential virus, then you want a cross-reactive vaccine, something that has a strong cell-mediated response, is able to properly present conserved areas which don't mutate readily between variations of the virus, and are able to be protective over time and over mutation.
Now, since the virus changes and adapts to different types of weather, do you think that there could actually be a better time to administer the vaccine because our climate and the weather could allow it to work better? For example, more natural social distancing, not during flu season, that kind of idea?
Certainly possible. That's an interesting thought. I think that, I mean, in any one situation, there are so many variables that have to be taken into account. I think it would be very difficult to make a call one way or another until you could really sort of look at each one of those in isolation and also collectively.
We're going to talk about a vaccine now, Rachel. It's something that Canadian researchers and researchers around the world are working really hard on, right?
Oh, absolutely. And, Chris, over the past few weeks, I've had a chance to talk to so many interesting researchers. I was talking to the University of Alberta in Edmonton, and they said they have over 10 projects on the go right now that are just looking at vaccines and the best approach. So, Roderick, whether it's your team or a team in Canada, a team around the world, I know so many people are working on this. Are you confident that someone out there will be able to find a vaccine, something that's successful, something that works?
The way I see it, first of all, is that this isn't a race against other groups to find a vaccine. This is a race against SARS-CoV-2 to find a vaccine, right? That's really the end goal here. And even though one may prevail and get there's a lot of science that's going to be garnered from doing this process. There's a lot of great experimentation that's going to work toward furthering more beneficial vaccines in the future. We are doing everything from very traditional approaches to vaccines, which makes sense because they've been validated and maybe a quick way to generate a solution, but may not be the most efficacious or have the greatest durability. Everything from there to more experimental types of vaccines, which perhaps haven't been validated before and are coming more from the oncology realm and immunotherapy approaches that have been used in that realm, but maybe far more efficacious downstream. We're going to learn a lot from this process, that's for sure. And I think it's going to really set a stage for how we can model new vaccines in the future as well.
Qasim and Roderick, have you ever seen so much thrown at a particular problem in the past?
No.
No, me neither.
No, never. I've never seen so much work happen around one infectious agent in my life. Yeah, that is a first for me. But I've never been alive during a time when there has been such a sense of urgency.
How exactly do you grow the vaccine?
It depends on what your vaccine is. So a vaccine is it's just a one word, but there are many different kinds. It could be anything from nucleic acids, whether mRNA or DNA. It could be a protein and just a subunit protein, which is recombinant. That could be a generated form. It could be a virus-like particle, which has all the components of a virus, but doesn't have any of the genetic material. It could be a whole virus that has been inactivated and treated with formaldehyde or heat, or in some way killed it so that it can't replicate. It could be a virus that can replicate, but has been so dramatically attenuated that it's very weak and just can replicate very slowly so that it actually mounts a very powerful immune response in so doing. There are a lot of different approaches and strategies around this.
So a vaccine goes in and it stimulates your immune system to fight a predator that's at you.
Yes, that's right. We have different sort of branches of our immune system, and that's really where the basis of how to develop a strong vaccine is really based on understanding all of our immune system and how we can mount proper responses. And those responses have to be properly oriented toward the type of pathogen or what the type of immunogen is that we're trying to fight off. In the case of a virus, this is an intracellular pathogen. So if we can generate a type of vaccine that tries to at least mimic that same passage and production of these viral proteins inside cells, then those tend to sort of mount the right type of cell-mediated responses that are the best at clearing those.
I envision like a greenhouse, kind of a building with all these test tubes and everything, and concoctions. And is that what it's like, or am I, in comics?
There are a lot of different parts to it. So, I mean, if you're looking at our end, we are on the preclinical side. We're the strategists behind it, figuring out what that vaccine will look like, doing the cell testing to ensure we're getting the right interactions, to make sure that every aspect of that process is happening the way we would expect it to. Then there's the sort of scaling of it and the manufacturing of it or when we have obviously the animal testing component, the preclinical testing piece that has to go along with that to show that at least some kind of an in vivo model is working the way we anticipate it against the virus. And the immune responses are the way that we would anticipate them and expect them to be. And then we have to manufacture it and scale it up at a human grade level, which is far different than what we'd be doing at the experimental level. And then, of course, once you have that done, you start going into your clinical trials, and hopefully everything's working the way you're expecting, and you can show that it's safe and efficacious, and your immune responses are working, and you're not hurting anybody, and then you've got something that hopefully will work.
This is really fascinating. Listen, and this is why it's where you get Qasim and all of his research on the spread of COVID and how the weather may play a role into it, and then we learn from you that this virus mutates as it spreads. I mean, it becomes a really complex problem for science to try and deal with. Dr. Vanessa Meier-Stephenson is a virology researcher at University of Calgary. Thanks for joining us today. Let's talk about the spread of this. And Rachel's here too, because you were saying you thought initially that 2 meters wasn't enough. You thought maybe that you could catch the virus.
Yeah, it's interesting. It's how aggressive is that sneeze, right? How hard are you coughing? How hard are you spreading those droplets? So, Vanessa, what's your idea on this? Is 2 meters, that social distancing, is that enough?
Well, actually, the distance of 2 meters actually came from some of the original studies during the pandemic flu of 1918. It's actually from soldiers' quarters where the beds of the individual soldiers were packed so closely, and they noticed that whenever someone was sick, that anyone within that 2 meter distance tended to also become ill, and so that's where that 2 meter mark, that 2 meter distance came from. Of course, we certainly have a lot of new studies that are looking at various ways of measuring how strong a sneeze is, or how far you can project that little droplet of potential virus across the room. And various studies have shown that 2 meters is generally that distance that we need to be concerned about. Yes, there's the odd particle that can reach a bit beyond that, but for the most part, with any conversation, much less than that, of course, but a cough or a sneeze tends to be within that range. So I think we're all pretty safe still sticking with our guidelines of that 2 meter distance between each other.
I bet you there's a lot of people that now that it's allergy season, they might wonder, do I have an allergy? Do I have COVID? Do I have the flu? Do I have a cold? How can you tell? What's the difference?
Allergies, of course, are caused by your body's reaction or overreaction to certain allergens in the environment. So like pollen from plants. And for people that get allergies, they tend to have that sore throat, stuffy nose, itchy, watery eyes. And occasionally people will feel a little weak or unwell, but for the most part, that's kind of the limitations of those sort of symptoms. For some people with asthma, though, there is also the potential, or certain types of asthma, I should say, a cough may also be precipitated or provoked by certain allergies. So that's the sort of, you can already hear those symptoms that we talk about that do overlap. So that cough, that we tend to associate with the COVID symptoms list that we get. The stuffy nose, stuffy runny nose certainly is also in there with that. What makes it different with COVID, however, is that they also, people can also have the full muscle aches and body aches. So not just weakness and feeling kind of run down, but just full body aches. Also, some people can also experience trouble breathing, certainly the more concerning one, and fevers. So if you have fevers, trouble breathing, and that cough that is out of your norm would certainly indicate a possible COVID infection. And I say possible because there's still other viral infections out there that could do this. But for the most part, what we would recommend is that people still identify themselves to public health to get tested and try to arrange for testing, keeping away from other people while this is happening, keeping yourself self-isolated, because again, we don't really know for sure. It's reassuring if you're someone that's always prone to getting allergies in this type of season, that if you've got that itchiness to your eyes and that stuffy nose and it's been there for a few days and nothing has changed in terms of developing new symptoms. But because we can't be 100% sure, and the risk of exposing yourself to someone else if you may have an infection or have the virus is far greater than keeping yourself tucked away just for that time while we're still trying to sort out whether it is a true positive.
I would like to get your opinion on the study that was done in California, where they talk about air circulation, and they've used air circulation in the built environment to sort of flush out any potentially contaminated virus particles inside a home. Now, for someone with allergies, of course, that may not be a good solution, bringing a lot of pollen and allergens in their home. But what do you think about this idea for people who don't suffer from allergies and want to just make sure their indoor air is cleaner?
So I guess the question is, are we worried about bringing virus into the house from that sort of an introduction? And I would say that that's a very low risk sort of scenario. First off, the virus is actually pretty weak. It is vulnerable to a lot of different things like drying up, breaking down in the natural environment. The mere UV light that from the sun that this is shining down on objects outside. And so a lot of those things in combination minimizes the amount that's really going to be found in our environment and circulating in the air as an airborne sort of virus. And so the risk of acquiring an infection from fresh air, from the wind blowing around, unless there's someone standing outside your window talking and coughing and from that, increasing the amount that's there. But for the most part, the fresh air has so many benefits from that front, clearing out the stagnant air in your home. Not a bad scenario.
With fresher summer air coming into play, do you think it is going to play a bigger role for Canadians? I know boosting our mood, of course, but just naturally helping with this situation?
Yeah, mood is certainly, and mental health is certainly perhaps an undersold benefit of a lot of this stuff with releasing restrictions and being able to get out and move around and interact with people. Can't be undersold. Let's be honest. But absolutely, having this summer season and fresh air and being outside, all great for being able to tackle this and get through this whole scenario. Just the same, we can't forget that it's still people that are spreading the virus, and every handshake, every object that's touched with dirty hands has that potential to be a vector and impact someone else's day, life, so family. So that's an important thing to remember, that even though, we're out there and we get to do more and more things or enjoying ourselves, we still have to remember that it's still circulating in the in our population, and every interaction has that potential to create a new potential infection. And we have to just be careful about that, because we're not just interacting with that one person, we're also interacting with that person's contacts as well, their family. So if we pass it on to that person, they may have an elderly parent who is in that vulnerable category. They may have another member of the family with another health condition. And so it's hard to really say where we're getting it and who we could be passing it on when we do those sorts of activities.
Viral Weather continues on the Weather Network. Rachel Schoutsen joins us now, and together we are going to bust some myths about COVID-19? Soap and water and your
You know,
hands.
Yeah, that is going to be the easiest. Soap is everywhere. We have soap in our home. We wash our dishes with soap, our hands with soap. That will kill the coronavirus. And coronaviruses are actually fairly easy to kill. You just need to destroy that outer layer, that outer lipid layer, and soap will effectively do that. So we all know washing your hands for 20 seconds, singing happy birthday twice. That's what you got to do. Warm water works better too.
Okay. I see a lot of people wearing masks now and a lot of people wearing bandanas too.
Mhm.
So one may be more fashionable than the other. Which ones work?
So it took a while for Canada to really create a stance on if we should be wearing a mask or not. And, the word the N95 mask is thrown around a lot. However, finding these professional grade masks may be considered to be irresponsible if you're not a frontline worker, right? If you're not working in the hospital, save those for the people who need it most. Teresa Tam did say that wearing a mask of any kind will be helpful in reducing the spread. But what I think a lot of people don't understand, Chris, is wearing a mask is not really protecting yourself. There's no research that backs up that a mask will stop you from contracting COVID-19, but it could limit the spread of your droplets. Perhaps you may be asymptomatic. It will limit the spread of that getting onto surfaces. So again, it's that responsible thing to do.
Okay, what about the gloves?
The gloves. I hate these gloves. I hate them because COVID is not going to get into your body by seeping through your skin, right? And I think that these gloves give people a real false sense of safety. If you touch your face with a gloved hand or not, and you have a contaminated hand, you could still be passing this virus through. I think gloves are an interesting one. They've created a bigger issue when it comes to litter. A lot of people are not removing the gloves properly. They are not throwing them in the garbage. So it gives busy essential workers another task, and that is cleaning up gloves. And from the doctors that I've spoken to, gloves are really not necessary.
Okay, we have a minute and a half left. You have something interesting about copper.
Yeah, so surface transmission and how long a virus lives on a surface. Copper. Copper was that one surface that the virus lived the least on, only 4 hours. And it's interesting, Chris, because copper actually has properties where it sends out its ions. And, I'm not a scientist. I'm not really properly doing this justice, but think of it like this: sends out ions, and those ions can actually break through that outer layer of the virus and help to destroy the DNA inside. So what does that mean? It actually limits the time that the virus can be active on a surface. So copper is interesting. Why aren't all hospitals made of copper? It could be really good to decrease the spread.
But it's a pricey
And
metal.
it's expensive. It's an interesting idea that copper could naturally be one of those killers.
Rachel, thanks for joining us today on Viral Weather. Always good in weather and your health on the Weather Network with Rachel Schoutsen all the time. We also want to thank Qasim Bukhari from Massachusetts Institute of Technology, Roderick Slavcev from the University of Waterloo, and Vanessa Meier-Stephenson joined us from Calgary to talk about viruses today. We talked about where did COVID come from? How do we kill it? How fast for a vaccine? We do appreciate that you join us here on the Weather Network on television. Of course, you can download the podcast wherever your favorite podcasts are stored, Spotify and iTunes. I'm Chris St. Clair. Thanks for watching.